Welcome to the DNA of Things, where we explore how your potential is realized at the intersection of DNA, environment, and behavior. Today's guest, Hannah Dose, the co-founder and CEO of AU Genomics, a company pushing the boundaries of genomics innovation. From pioneering CRISPR research to winning XPRIZE for rapid COVID testing, Hannah's journey exemplifies how science can drive equality and accessibility. In this episode, we delve into her mission to solve high-stakes challenges, bridge gaps in health and science, and harness AI to transform humanity. Get ready to be inspired by stories that redefine what's possible in genomics. Hannah Dose, the co-founder and CEO of AU Genomics, a company pioneering bespoke, high-stakes, next-generation sequencing solutions in the genomic space, beginning her career with groundbreaking gene editing, CRISPR-Cas9 work in plant sciences. Hannah Dose shifted her focus to human health and accessibility in genomics. Her vision led to AU Genomics, and she capitalized on an impressive win at XPRIZE for her rapid COVID testing innovations, highlighting the team's work to drive innovation and dedication in making genomic tools accessible. As a 100% women-owned company, AU Genomics boldly addresses unmet scientific needs, and Hannah Dose and her team specialize in custom genomic solutions, from novel assay to development to maternal health projects often deemed too risky. Her journey and mission represent a new biotech model, or tech bio model, grounded in expertise, innovation, and a commitment to advancing science and accessibility. Hannah Dose, welcome to the DNA of Things.
Thank you. Thank you. It's a pleasure, honestly. I'm very, very happy to be here. So thanks for having me.
My absolute pleasure. We were connected through Chris Barbizet, who we had on the show recently, and he said just absolutely great things about the work that you're doing. As a fellow molecular biologist myself, I know the excitement and the, let's just say, daunting nature of the bench. And it sounds like you're quite masterful in the wet lab solutions, but you're also bridging with digital technology. And so before we dive in on some of the stuff that you're working on, to the extent that you can share, I would love to know the personal side of this. So at the DNA of Things, we're about high tech and high touch, and really interested to learn about the human element of your story. So how did you get to where you are pioneering these risky technologies in the next generation sequencing world?
Yeah, so that's an interesting question because it can get a little tough to pinpoint exactly where I decided to go down this path. I think I've always been kind of a futurist at heart, big sci-fi fan as a kid. And my journey really, it's a bit different than what most people would expect. Typical path is generally, getting PhD and going down that route. And, I didn't do that for a few reasons, and thinking about it, maybe I was also a little bit impatient, but I'm definitely more of a trial by fire, learn by doing, just kind of like get in there, get your hands dirty kind of person. And so I try to find a way to immerse myself in what interests me and problems that I see, and how to find solutions for those problems. So, gosh, I mean, I guess the first kind of intro to this was at university. I studied both astronomy and plant biotechnology at the time, and I really wanted to design plants to improve actually agricultural production for long duration space travel. That was always a dream of mine to do. And this was in
Mm.
Hawaii, which was actually kind of a perfect place to be at the intersection of those two fields. You have Institute for Astronomy there, you have the NASA Astrobiology Institute wing, you have all the agricultural plant research going on. And so I really focused mainly on plant genetics, which at UH, it's deeply rooted in plant protection,
Mm.
self-sustainability, ahupua'a systems. They're a big part of Hawaiian culture and the essence of community. So To keep things short, I got really into plant science. We were using CRISPR to create crops that were resistant to certain mildews. And basically, we focused on specific proteins that were released by pathogens, and they would allow the pathogen to actually insert themselves into the plant. Now, there were also proteins that were created by the plant itself that kind of conferred that host pathogen interaction to allow the pathogen to enter. And so with CRISPR technology, what we did was we targeted we targeted the proteins that those effectors kind of used, those proteins from the bacteria. And by knocking out those proteins, the mildew actually was not able to enter the plant. And so that was some really cool work. But through all that, my mind was really feeling like all of this research was built upon one main idea, which was understanding the genetics of these interactions. And so everything we did was based on DNA sequencing. And so there was really a spark there that caught my attention. I had a growing fascination with this immunology of the plant, essentially, and these cell signaling, and the interactions that would happen between the host and pathogen. And it got my mind thinking more about how, immunology works in humans. I know a lot of times it's the reverse way, but starting with the plants and kind of just opening my eyes to using DNA not only in the sense of diagnostics, but using it as a tool to kind of to be able to go in and change how our bodies can interact with, you know, external factors. And so with that newfound, kind of excitement, I joined La Jolla Institute for Immunology, which is out here in San Diego, California. They're actually one of the top immunology institutes in the world. And I started with working in the DNA sequencing core. Now, this was right before COVID hit. And quickly, I kind of moved up and started running the core. And so when COVID hit, we were doing a lot of projects with vaccine research in the institute. However, really early on, we still had to shut down for a couple weeks like everyone else, even though there was research going on that was really important to vaccine development. And so the institute kind of asked us, hey, can you find out a way that we can test whether or not our researchers have COVID so that they can come back to work and be safe and continue to develop vaccines and do more research on COVID. And so we jumped at the opportunity. We already had a lot of sequencing data from positive patients. And so we actually developed a rapid COVID test that was saliva based. So it's non invasive, very, very fast, and it was high throughput. And there were a couple of main benefits to this test. One being that at the time, testing really was not a thing that a civilian can go out and get and purchase.
Mhm.
It was extremely cheap. So I think it was about down to $1.29 per test. And the fact that we were able to do this at scale, so we could run thousands of these a day, it was really, really important that we were able to make this public. So that was our main goal. Once we developed this, was to release it so that anybody anywhere in the world could do the same testing. And at the time, there's XPRIZE, who is an organization that they create competitions to kind of incentivize beneficial technology advancements for either humans, for the environment. Like currently there's a carbon scrubbing one, there's a test for longevity. And so at the time they released a COVID testing competition because it needed to be cheap, it needed to be fast, and it needed to get everywhere in the world. And so we actually we applied for the competition, and gosh, I mean, we did so many iterations of this. We ended up removing extraction. We ended up being able to remove a lot of steps to bring down that cost and make things faster. And we ended up winning the competition, which was awesome. It was a $6 million competition. We were one of 5 teams that won. So the La Jolla Institute for Immunology got $1 million for this. And with those winnings, I think this is the important part that kind of fueled this step into equitable health, is that with the winnings, during the time we worked with, actually, there was a PI at La Jolla Institute, Sujan Shrestha. She is from Nepal, and she has a lot of contacts with the Nepalese government there. They do a lot of research together. And she notified us that Nepal really did not have access to testing. And this had been months after, you could go to CVS at this time and grab a test, but Nepal still didn't have access to this testing. And so she was like, hey, you really need to get in contact with them and see if there's a way that you can help them out. So we actually were able to donate a year's worth of testing reagents. We had an 8-hour long Zoom call with them, just kind of going through exactly how to do the test and replicate it so that it was the same every time. And yeah, so they were able to get COVID testing. It was saliva based. And not only that, but we were also able to donate COVID sequencing reagents to them, because at the time, research in the changes and variations of COVID, this was right before Omicron, there was still a lot of research that needed to be done in the world. And so Nepal was able to join the global conversation of COVID tracking and understanding with the testing reagents that we were able to donate. And I think importantly, through that, it wasn't necessarily, okay, they needed it and we donated it. There was a part that we realized and we learned when talking with Kathmandu University out there, was that they actually could not get the tests past the border or get the sequencing reagents past the border. And it kind of opened my eyes to realizing how difficult it is to kind of bring technologies to other countries, not just because of financial reasons, but there's also issues with, with importation and how who on the Nepal side has the clearance to accept these and then distribute them. And so even during the time, this was an Illumina test and it hadn't been released yet, we kind of got the beta testing on it. And we talked with Illumina. We're like, hey, you know what? This lab really needs these tests. Like, how about we'll pay for them? Just go send it to them. And they're like, we can't. Because we are Illumina, we are this company, we have certain people that can accept these, and we cannot actually send them out. And so it was a weird situation, and we were able to find that loophole to kind of just get them out to them. And that just really that was the first time that I realized that it's not just an issue of, whether they have the money or whether they have the foundation to be able to run these things. They had a sequencer. That wasn't the problem. And so, yeah, so it was just one really big aspect of, okay, health inequity across borders is even more drastic than we even see between communities in the United States. And so after that point, we continued with our work at the La Jolla Institute with our next generation sequencing. We were bringing in new technologies to the Institute, some really big things. Single cell became a really, really big thing the last couple of years. or last few years. Spatial sequencing, we started getting that up and running. We now have a nanostring out there. We were doing 10x spatial sequencing. But one of the really important things that we were doing is we would do method development. So somebody might have a scientific question that has never been asked in this specific way before, or they have a weird assay that hasn't been tried, or they might be working with a really difficult cell type. And the difficulty with that is, you can spend tons of resources trying to, start from the bottom with, okay, how are we going to put this together? How actually does this look on the bench? And so we, as our, main kind of work there was being able to do this method development for researchers. And so we would actually often get times where an external, lab or a company or something would come to us because of our method development, and they would, need some work, but the institute ended up having to kind of shut down that external work because they said, we need to focus on internal projects. And unfortunately for them, they always came back and said, look, you need to help us because nobody else will take this project. These samples are too difficult. This design is too difficult, or it's very risky. And so that kind of was the birth of AU Genomics, because these researchers really had nowhere else to go. And so we had the skills, we had done these kinds of things before. We're really scrappy. We make stuff work. We have dealt with a wide range of very difficult problems. And so we kind of know how to tease these things apart. And that's one thing we're really good at. And so for AU Genomics, one thing, or our main vision really, is to be able to bridge the gap and bring accessibility to these difficult, not necessarily even technologies, but difficult methodologies that people might use in creative ways that, have never been done before. Some examples of this might be even hacking a sequencer to create an assay, a large scale, high throughput assay for, actually, maybe I can't say exactly what it's for yet, but yeah, just kind of going beyond what a typical, whole genome sequencing project might be. And our goal really is to not just be a service, but be more of a partner, be more of a collaborative partner that, if you might not have the access to these resources, you might not even know where to start. You might be a data analyst, you might be a family member of somebody with a rare disease that, you have some background, and you really want to explore different areas of how the physiology of this disease might affect other patients. You're going to need a whole team and resources to make that happen. And we kind of put all those together for our clients, whether, you're in ag tech, whether you're in human biology, whether you're in drug discovery. We're able to kind of make things happen that haven't been happened before, or haven't been possible before. So that's our goal. That's the journey. And I'm really just getting started. The AU Genomics has been around for 2 years now. We just hit our 2 year, actually, no, we're about to hit our 2 year mark. So we're quite new. And, like I said, we're scrappy. We're making things happen. And yeah, we're really excited about the rest of the journey.
Amazing. And how big is the team right now?
We're quite small, so we have about 4 of us on our team currently. Yeah.
Wow. So you must
Wow.
all, big thinkers, and it sounds like there must be a ton of synergy going on there. Like 1+1+1+1 equals a trillion. Because to take on
Right.
those projects globally and to be able to provide timely solutions under such urgency is, well, it's just amazing. No small feat. And, really interesting journey. I didn't want to let it go without asking, because I'm a science fiction fan myself. What was your favorite sci-fi movie growing up?
Oh man, I'm a big Star Trek fan.
Okay.
So I used to want to be Dr. Leonard McCoy Bones. That was
Burn.
what I thought that was gonna be in my future. Couldn't convince me otherwise. But also I'm really big into novels as well,
Mm.
sci-fi books. So Three Body Problem is a big one that I read as well. Yeah.
Yeah. One of my
How about
favorites.
you?
One of my favorites is 2001: Space Odyssey.
Oh
Um,
yes.
right. Well, the
Yeah.
origin of everything, right?
Right.
Um,
Right.
but remember, I don't know if you remember Gattaca with Ethan Hawke.
I was gonna say, yep,
Yeah. There's no gene for the human spirit. We'll
yep.
see. We'll see. Right.
Right.
Yeah.
Right.
Yeah. What else? Well, you've probably seen Martian. I mean, given your interest in, plants for outer space with,
Right. Yeah, Marston.
and I don't know if you'd consider like the Marvel series sci-fi, but I mean, it kind of was when you got into the Infinity Wars. I mean, gosh,
It's true.
a little different. I think the sci-fi is different than superhero, but you mentioned Kathmandu and Nepal, and I started thinking about Doctor Strange. So that's the Doctor Strange series. He goes to that region, but yeah, it's amazing how many scientists I speak to that have that kind of almost like artistic, creative origin that go on to be researchers and entrepreneurs. Yeah, I really got to study that a little more, see if there's any significance, or I'm just biased in remembering the stories that are most unique. But yeah, this theme that you've been touching on, like you talked about plant bioscience and CRISPR-Cas9, and I just want to understand this correctly. So it was knocking out a gene that made a protein that the pathogen of that plant mold would eat normally. Okay, yeah, yeah.
Yeah, not necessarily eat, but would use to be able to enter the plant. Yeah, so it's
Got it.
kind of like a signal that
Yes.
the bacteria would use to be able to like, okay, here it is. Here's the entrance. I'm getting in. So,
Right.
yeah. Mhm.
Gotcha. So a signal, not necessarily receptor, like what we're seeing in in the HIV, the story that you heard of those twins in China, right? The
Right.
doctor that went to jail for applying CRISPR-Cas9 to knock out that gene variant in the T cell receptor for HIV. Yeah. But what you said, made me reminiscent on some of my, academic studies that were looking at like the coral hollow biont. So there's a researcher based in Israel, I think Eugene Rosenberg. He proposed this whole idea of the coral hollow biont. So these healthy coral living harmoniously with this coral microbiome. But there are invasive species there, invasive species of bacteria. So there's this whole kind of corrective approach using microbes, for example.
Yes.
And yeah, the same is true for gut microbiome as well. But really
Right.
interesting, like what you're talking about, you're alluding to is like the crosstalk between species, right? So,
Mhm.
higher level organisms and more simple organisms. I'm not a big fan of calling microbes simple. I think they're actually very progressive in terms of what they can do. But yeah, like seeing things like, transcriptome profiles in, human epithelial cells or even brain cells being turned on and off based on what signal molecules microbes are making. So really fascinating to hear you doing things other than, just creating a bunch of sequence data, which is obviously very important as well. But the fact that it's about this kind of repurposing of technologies, like you talked about. CRISPR-Cas9 is essentially this viral immune system, let's say, for the sake of simplicity. But now we're taking it and doing very targeted base changes in in plants and well, in humans too, right? Because there's the sickle cell therapies that have been developed.
Yep.
Um,
Yep. Exactly.
yeah.
Exactly. Hey,
So
I
can you?
was also,
Sorry.
oh, sorry.
No, no, no. Please.
Also, there are some interesting ones in oral health. I'm not sure if you had been following. Yeah, so there's some, let's see, I think it was, I want to say GTFB genes that are responsible for the formation of plaque biofilm, which
Yeah, okay.
if if for those of you that don't know, just like breeding ground for bacteria that cause periodontal disease,
Right.
like streptococcus. Streptococcus mutans, which, or oral health is a whole other crazy world. But yeah, so they're editing that gene to reduce the generation of those plaques, and that in turn reduces the bacteria that grows. So that's really interesting.
Which in turn, in theory, would reduce, heart disease, because like a lot of the periodontal issues, basically these invasive bacteria can colonize your arteries and create issue there as well. So it's actually interesting to think about it like, oh, well, you're genetically predisposed to heart disease, because of these, specific genes related to, say, hypertension. But then, oh, actually, because you have genes that make you prime breeding ground for bacteria. It's a bit of a paradigm
Right.
shift, because we don't think about ourselves as being invaded like that. We like to think of our insides as being relatively pristine, but it's not the case. I actually did a lot of microbiome work with Ruth Lay when I was doing my postdoc at Cornell, and we looked at, basically plaque biofilms. Yeah. And, also did like how the infant gut is colonized over time. We looked at how the gut microbiome is related to the human genome. So there's, definitely some, genetic signal in humans that would suggest a specific kind of bacteria being present or absent. So exactly what it is that you're talking about. And, with it feels like, say 10 years ago when I was doing that research, or longer, it would feel like it's such a daunting, pursuit to look at all these inputs and outputs. Like, how do you even measure them? Like, you talk about all the positive and negative synergies possible, but now well, AI is not new. I mean, you think about like the way that it's more accessible now with
Mhm.
large language models. Certainly, that's new, and there's all kinds of data structuring and organizing. I think that's great. Like, I think back to my PhD days, I could probably do the analysis that took me, let's say, a year, probably do it in a week with all the tools now. But I still had to, have the experimental design, and I still had to think some things through. And we use a lot of, phylogenetic algorithms that are, maximum likelihood. They are machine learning algorithms, basically these tree building algorithms that look for a signal, and if it goes the wrong way, it'll tear down and rebuild. So, yeah, not really new, but the scale of accessibility and the generative aspect of it, I think, is really exciting. Are you seeing that? Like, I mean, I know you've only been around 2 years, so it's almost like you probably didn't know a world without AI, but do you see it, creeping in? Do you see, it changing the kinds of projects that you're getting? And what projects can you talk about? Because I know some of them are super stealth, but besides like COVID, what are some things that you're working on that you could share?
Yeah, so kind of to speak more on the AI side of things, one really interesting thing that I've discovered is there's actually a lot of companies that are generating algorithms, and they need more data for it.
Uh,
I mean, I know that there's, biobanks out there with tons of data, but more of the metadata that is involved that's, maybe either missing, maybe either they don't know how accurate some of it might be. And so we've actually had companies that kind of are made to create these algorithms come to us to be like, hey, do you have some data we can use? Or let's do a project together. We specifically need to focus on in this subpopulation or specific disease area. And it's interesting because it seems like there's going to be a lot more data that is gonna need to be generated. I know, like I said, there is definitely lots of data out there, but I think, gosh, what are we approaching? There's something like there's gonna be like 40 billion gigabytes needed or something of genomic data out there pretty soon to kind of answer a lot of these questions. And so that kind of is one aspect where we definitely see it creeping in because it's kind of like sometimes there's a solution and they're looking for ways to plug it in.
Got it.
And we can kind of be the liaison for that. in a few situations, like with some of the relationships that we have with either healthcare centers or researchers that are, within a hospital, kind of focusing on a disease area. So that's kind of one interesting place that we found ourselves in that I didn't really expect to be in when we first started. I would say, yeah, to yours, AI definitely was a thing. I think in the beginning there was this unknown of how far it was going to go. I think there was one side that was like, okay, we've solved all the world's problems now. Like, we're about to know everything. And then there's the other side of some people still reluctant to use it. Some people maybe felt like they were getting bashed over the head with the term AI, in kind of everything that they were doing. I know there were a lot of companies that there was a period of time where for getting investments, you kind of had to integrate AI, and it was like, okay, how are we going to do this? Because this is what investors want to hear. And so yeah, it's definitely a big part of what we do. I think, interestingly, for me in the space that we're in terms of accessibility to new technologies, I mean, we certainly embrace it. And the importance of this is not only in the insights and being able to get these insights faster, but also in cost too. And kind of the drive for AI itself has kind of, it's piggybacked, or in the scientific kind of innovation side has piggybacked on the fact that a lot of new, if you think about computing side of things and how much further that has gotten because of AI as a whole, not necessarily just because of genomics, but looking at, some of the advancements. I mean, NVIDIA, the work that they've been doing, I think one of their GPUs was like, gosh, there was one thing I saw, it was like there was a big study that with 30 hours of CPU Amazon EC2 computing was like 25 minutes on an NVIDIA, the A100 GPU chip. And so being able to kind of speed that up, but at the same time making it cheaper, because, the cost time, I think it was like a 90% price reduction during that study for what was previously done on the 30-hour chip. And so what some people might not realize is the majority of sequencing costs is not exactly in the sequencing itself.
Mm.
A lot of that cost, comes from, well, you have sample collection, but also the data management, data processing, secondary analysis. Those things can all be very, very costly. And that actually eats up the majority of sequencing costs, um, at least in the past. And it looks like things are changing. And it's it's kind of interesting because when you think about like Moore's law, I saw something about this recently. I think it might have been, a year old, which is old at this time, but Moore's law, where, the number of transistors on a microchip doubles every 2 years or something like that. And then the converse of that, they were calling it, well, I guess it would be eorms. It was
The
the
reverse Moore's law. Yeah.
reverse, yeah.
Yeah.
Yeah, and it's the idea that, developing a new drug tends to cost twice as much every, I think, 10 years or 9 years or something like that,
Mm.
which is really interesting because you have these two opposite things that somehow can exist together. But I think that the increase in use of AI can actually start to reduce that Eroom's law, because, a lot of the cost of that is being unable to predict whether or not a drug will actually be successful.
Right.
You know,
Right.
we there's like 90% of failure rate, or maybe
Right.
even higher, something like that in drug discovery, and a lot of that can be be lessened with the fact that now we have these prediction models where, I think, trying to think of some, DiffDoc, I think, was one of them for drug molecule binding. AlphaFold, I know, is a popular one. And then like MoFlow for small molecule optimization. These things can all really be applied and really reduce those costs so that we're not just, throwing money at something either an incremental benefits to something that already exists, or to overestimate whether or not something is going to work. So I think it's plays a huge part in all of this. That's a little more in the drug discovery side, but yeah, it's definitely been interesting to watch. And I think for us, the sooner that we can kind of implement some of these ideas is going to be the better for us and the better for research in general. I think some of the difficulties there are the fact that we don't necessarily do the same thing every time, all the time. We're always doing, different wild projects. And so building those pipelines in the first place still takes some time. So it's a little different for us, but I think being able to kind of pivot when needed and pull in the right tools is going to be really important for us.
Mhm. One of the things you mentioned at the top there was, different organizations reaching out to you, looking for specific data sets, right? Like, so we know, for example, that it was like the UK Biobank, extremely biased data
Mhm.
set, enriched in Northern Europeans, I think even mostly men, if I'm not mistaken. But, you
Mhm.
see the emergence of things like the All of Us initiative, where I think it's
Yep.
10 million genomes and, set of diverse backgrounds contributing. Do you do any work with those guys?
Not in that sense. I'm a volunteer in
Yeah,
in
yeah.
there,
Like
but
folks can sign up and be like partners on the. Oh, you're a volunteer, like you're part of the study, kind of thing.
yeah, yeah, I
Cool.
jumped at that. I was like, heck yeah.
Yeah, it's really
But
cool.
but yeah, we haven't yet. That's something that we definitely want to do as things are moving along. I think, you know, being a part of that is going to be increasingly important because the actual people doing the sequencing, there's really not that many resources out there for the actual sequencing side. There's lots of, people ready to participate. There on the other side, there's lots of people ready to kind of digest this data and get into it and train their models. But the sequencing portion itself is surprisingly a pretty big bottleneck. So us
Interesting.
being able to be a part of that would be
Yeah,
definitely important. Hmm.
yeah. Well, I'll see if I can get Joe Handelsman to listen to this podcast. She's one of the advisory board members there. Actually worked with her postdoc way back in the day, Pat
Awesome.
Slosh. Anyway, I think she's a White House advisor back in the Obama administration, but she's one of the advisors on the All of Us project. Yeah, I mean, I think I haven't followed up in a while, as in a couple of weeks, but I understood that funding was an issue with
Hmm.
that project, and it's such an important work. I really hope that becomes prioritized. One thing I'm really interested in particular, and I shouldn't really speak out of school because I haven't really thoroughly analyzed this. This is just kind of a knee-jerk reaction that might be wrong, but I'll go down it anyway, and maybe you can correct me. But when one of the first publications came out, I saw there was this figure on it was like a principal component analysis, and the data were structured or colored by race. And I thought that was really interesting. And now I was reading Stephen Jay Gould, The Mismeasure of Man at the time.
Hmm.
And so, it seemed to me like an odd classifier to put on a figure, race. Like I could understand something like skin pigmentation or geographic location or something more biological. So again, I have to dive into what was really intended with that. Maybe I misunderstood, but even the thing that's interesting when you start to think about how, say, I identify my race. I did an interesting analysis of different markers involved with like human performance, right? So
Mhm.
genes involved in exercise response, genes involved in nutrition response, and genes involved in recovery, right? And then I looked at the geographical, um, the allelic frequencies, the geographical allelic frequencies based on those markers. And so while I may be Eastern, Northern European with some Viking DNA in there, actually these functional genes would say, well, you know what, you train like a North African, you recover like an East Asian, and you eat like a Mediterranean.
Just
So, I mean, and that's to say that, our genomes are very cosmopolitan, right? Like
Mhm.
we're not isolated in these geographic re- geographic regions by geographical barriers, let's say.
Well.
So I think the way that we start to cluster data becomes really, really important in the future. Like some of the work that I did, this is now more in my PhD days, we used multi-locus sequence typing, which in this species of bacteria, archaea, that live at like super high salt concentrations. And it's basically such that they're the only species of bacteria that live there. But we looked at these like specific, again, these loci in specific genes, and you look at housekeeping genes, which are generally more conserved, like things that are important for cell division or transcription, et cetera. And then you would look at more metabolic genes, which may not be. There's more flexibility for mutations there. And what we saw when we took single-celled isolates and basically analyzed a collection of loci per bacteria, what we found is they weren't clonal rates of population expansion in these regions. They were actually showing recombination rates at a higher frequency of sexually reproducing organisms, because the cells are in the environment, they break, and other cells pick up the DNA and it recombines. And so, anyway, that is to say, on these things that really look the same in terms of a single cell, they're very, very, very different. And so, human evolution is, I mean, it's a very complex thing. It's a very touchy thing too, right? Because if you think you have a certain heritage and you learn something different, that's hard because you identify in a way with your ancestral origins. So anyway, all that aside, one of the things that you're saying about, well, it's so expensive to develop drugs. It's like, well, I think maybe the drugs are serving like a greater diversity of people. And like, take, for example, the gene knockout therapy for, people who are at risk for accumulating those bacteria that cause plaque.
Mhm.
I think Streptococcus mutans, what you said.
Mhm.
Well, not everybody would need that intervention, right?
Well. Yeah.
And so then not everybody would need to go into the trial to develop that drug.
Mhm.
So, I mean, so you can start to imagine, you know, a different way of assessing risk in a way that is much more objective and quantifiable. And I mean, that's some of the, the work that Mike Polcari there is doing with like haplotype is, the collaboration with pharmaceutical companies to, if you want to develop drugs for, say, hypertension or cardiovascular disease, well, why not select your samples or partition your samples based on these different polygenic risk scores? And they're not complete, but maybe this is 10% better than the way that we're doing. And so does this get us to a better outcome more quickly? So it's a really interesting time, but I remember this when all the sequencing was happening. My honors thesis supervisor, Dr. Melanie Dobson, I think she might be the first, I think she's the first Canadian female Rhodes Scholar, first human to see the Y chromosome sequenced. Anyway, I did my undergrad work with her, but I remember, before starting my PhD and going into more metagenomics and sequencing, et cetera. And she's like, somebody's gonna have to like actually do the biochemistry to analyze the effects of all that genomic data. And so there is that kind of, it's like we
Mhm.
didn't know what we didn't know. And now it's like, wow, there's so much work to do. It seems like there's, room for improvement on the computational power, but room for improvement on the assay development as well in terms of, the economies of scale. So,
Yes.
so look, I mean, it's it's really amazing to hear how you started with, the sci-fi, building plants for interstellar travel.
Right.
Now you're like, making genomic tools accessible. You're solving hard problems. I'm interested to know like what your vantage point is like now versus, that, bright-eyed graduate student. Now you're this, trailblazing entrepreneur. What has, changed for you personally, and what are you like really excited about for the future?
I think probably one of the biggest things that have changed for me personally is realizing that I can make shit happen. I think that was something, my bright-eyed early self. I think a lot of scientists go through this in your early career, is just feeling like, I don't really know that much and everybody else knows a lot more than me. So I'm just gonna, just kind of sit back and be quiet and learn as much as possible. And I think kind of taking a leap in the beginning of this, I think, especially the XPRIZE, all that kind of spearheading all that work, it was a really big part of the journey, just realizing that I made this happen. This was an idea, and now it's a reality. And getting the right people together to support me in this, having the right, whether it's mentors, having the right connections of people to kind of fill the gap of things that I don't quite have, but I know that I need, and I know how to express what I need from it. That was a really important thing for me personally, because it made me realize that I don't need to have all of the answers for everything.
Mhm.
I need to surround myself with people that know tons more than I do in a very specific thing, and they can help build this, and just to keep the overarching kind of goal, that end goal of, we're going to make this happen, making that possible. It's not going to be done with just yourself. So I think just being, yeah, just making things happen was more of a reality than what I had realized kind of in the beginning. And what that looks like for us, I know I kind of touched a bit on equity in access to healthcare. I think a really big part of that is also equity in access to science itself. Um, a lot of times, we're talking about race and your different experiences that you might have that kind of build up who you are. And I think also psychologically that can change the way that science happens. People have different experiences, they come from different backgrounds, they might have a completely different perspective on how to tackle specific problems, or they might, see more of an issue, whether it is in a marginalized community that might not get the advocacy that they need, or whether it's a scientific issue, like more of a molecular issue, that they can just have a different kind of background to bring to the situation. you know Being able to make these technologies more accessible. You don't need to have your own lab, you don't need to know everybody to make this happen. That's what we want to be there for. We can kind of power your ideas. Somebody that might have, an idea for something that's never been done before, we can assess the risk. We can put those building blocks together and make it happen. So I think, yeah, so long term, I kinda went on a big tangent there. It's just kinda sometimes I do, but, yeah, just for AU Genomics to be able to kind of give a voice to people that don't have the resources otherwise, and give the kind of playground to, you were talking about creative thinking. Somebody that might come from more of a creative background and, maybe didn't just follow the path of the typical scientists that, now they're a department head and they can try all kinds of experiments. Somebody might have an idea before that point. And so us being able to let people kind of play around with these ideas and really kind of tease apart questions that have never been asked before. That's what we're here for. It's interesting because it's not like, only people from top universities or only people from companies that have tons of money are ones with good ideas.
Mm.
Um, Lots of people have tons of great ideas all the time, but they just might not get the chance to kind of put it into action, to test it out. And so I think the more times that these maybe sometimes off the wall kind of theories and theoretical ideas that can be tested, if there's a path to test it out, we're going to be a lot better off having done so. Also, considering the risks, but
Mhm.
yeah, I think that's one really important aspect of us being able to allow, people
Mhm.
to really experiment.
Right. Well, that segues nicely into the preamble we started with at the top of the call, which is, you know, if this was your last podcast, what's the message? And that's number one. Number two is, who needs to hear it, and then number three is who's our next guest on the DNA of things.
Okay. I would say that, geez, I want to say a couple of things, but if I have to pick one, it's to recognize how big of a gap realistically there is when it comes to genomics research, whether
Mhm.
that is from across borders. I know I kind of touched on that
Mhm.
briefly, whether that's, you might not have a background specifically
Mhm.
in a genomics lab, you might be a data scientist. I think I mentioned a parent that you might just not have the resources. You might have a startup and, you're running off of not much, but you really need that one data set to
Mhm.
really drive things home and get to that next step so that you can commercialize.
Mhm.
There's tons of gaps out there. And who I really want to hear this is mostly, I think, I would say maybe investors is a big one, but also in governments. I know I didn't really tell this story yet, but just really briefly, in because we do some work with Mexico. Actually, one of the people that we work with is Daniel Uribe, who I know that
Daniel,
you
no,
had on.
Daniel's been on the
I
show.
said
Yes.
to that one.
Yep.
Yeah, and you know, actually, it's actually very interesting because the gap in resources there, it's not only, the GDP and issue of finances of the country, it's that when things are imported, there's one person that controls who gets that, and they set the price.
Mhm.
So people are left kind of at the will of this one group that can say, how much things are priced. And realistically, they cost more in US dollars there than they do here in the US by like threefold. And it's crazy, and it really stunts scientific research. So I think the government needs to really, really take a hard look at, how we can help to kind of just, I mean, obviously fix those issues, but stimulate scientific
Mhm.
research, not just Mexico, even in EU, this happens, Nepal,
Mhm.
Malaysia, all of, um, so yeah, that's
Got it.
one important thing. And then in terms of one person to be on, we talked about metagenomics a bit, and I
Mhm.
think it'd be really interesting to have Rob Knight. I don't know if you are
I know Rob
familiar
Knight.
with.
Yeah.
Oh, you do
Yeah.
know him.
Yeah.
Oh,
We
you've done.
sorry, go
I was just gonna say we've done work with him, and yeah,
ahead.
they're doing
Yeah,
some cool
Rob
stuff.
Knight. Actually, we collaborated on a paper 10 years ago, 15 years ago. Chime. It was the genomic analysis tool that we used to analyze all of the infant microbiome stuff. So that was back in my days when I was part of
Cool.
the Human Microbiome Project. So, okay, that's great. What a great trigger on the memory there. I'll definitely reach out to Rob. So just finally here, Hannah, what's the best way for people to find you?
You can find me on LinkedIn. Hannah Dose is me. I'm also on, well, my email, I can throw that out, is hannah@augenomics.com. And then also, let's see here, we also have a Twitter, but if you want to find me directly, those two are my main channels. Our Twitter is AU Genomics @augenomics.com, or X, sorry, or X is.
Yeah.
And
Don't offend Elon. You might, be building his like outer space crops.
yeah, that's not what I meant. Yeah, but those channels, whether you have a crazy idea, whether you just want to talk something out and talk about, the risks of something that you want to test out, and how much something realistically will cost, what it will look like. We also deal a lot with working across borders. So if you're in an area that, you need sequencing, maybe not even just sequencing, you need more of a partner, a collaborator, a holistic kind of approach to your experimentation, find us, and please reach out. We are more than happy to help. This is what we're here for. And yeah, thank you so much for having me. This was awesome.
Oh, Hannah, thank you so much. Yeah, this is gonna have a ripple effect. Thank you so much for your time and everything that you do. We'll be in touch soon.
Great. Thank you.
Thank you for turning in to the DNA of Things. Today, Hannah Dose showed us how science and innovation converge to break barriers, solve complex problems, and empower the underserved. In this episode, ideas were sparked and change is inspired. And if you felt that, please share it with someone who needs to hear it. Together, we can amplify stories that matter and bring the future closer to everyone. Don't forget to describe and subscribe and leave your review. Your support fuels the journey to unlocking human potential. Look forward to our next episode where we will be exploring more stories and the stories that transform lives at the intersection of DNA, environment, and behavior.